So now we will see transcription process, the actual process of transcription. Transcription means what? Basically, from DNA, the mRNA is formed. Means mRNA carries the information from DNA. The process is known as transcription, right? Again, I am repeating this. So from mRNA, the protein is formed, and this process is known as translation. So first we are completing transcription, then we will see translation. So this overall process, the overall process is known as central dogma. Okay, central dogma, the overall process. So let's first complete the transcription. Transcription, okay, for the transcription to take place, it requires a DNA strand. Okay, so from DNA strand, the mRNA is formed, which is going to carry the information, that is the genetic code from DNA, from the template strand of DNA. So let's discuss again in details now. In earlier class, in previous class, that is in the mRNA part, we have discussed in some basic things, and I left some point. Okay, so here I'm going to complete it, so you can add that point in your mRNA also. So we have DNA strand, double stranded strand. Okay, so this from these two strands, the strand which is going to give information, we call it as a coding strand. Out of these two strands, this can be coding strand or either this can be coding strand. It depends. Okay, it doesn't matter. So out of these two strands, either one of these strands will become coding strand. Coding strand means the strand that is going to give the information. Okay, so the strand which is going to give the information, that strand will become a coding, the other strand will become a template strand. Okay, template strand. So suppose this strand is coding strand. So for example, it can be this strand also. So let me write this one, coding strand. So if this strand has become coding, means it is going to give the information, the other strand will become a template. Become means what? We just give a name. Okay, template strand. Okay, so the template strand contain 3 prime, 5 prime. Remember, let me make it big. Okay, structure. The template strand is going to have a 3 prime, then 5 prime. Remember, I think in mRNA, the template strand I have given a 5 prime here. So you recorrect it, okay? You go back to mRNA part, and I think I have given a 5 prime here and 3rd prime here. Okay, so if it is so, go back and recorrect it with this. So template strand going to have a 3 prime and 5 prime. So the strands are anti-parallel. If one strand has 3 prime, the other strand is going to have 5 prime. If that strand has 5 prime, the other strand is going to have 3rd prime. Okay, because this is our coding strand. From this strand, the mRNA is going to carry information. So the other strand will become template, from where the mRNA is going to bind with it. Okay, bind with it. So here I am not going to show the nitrogenous bases. We have already seen in mRNA. Okay, remember one thing. The new nucleotide, that means, the new nucleotide, any new nucleotide which is synthesized, the direction is always from 5 prime towards 3rd prime. Okay, we have discussed this. So when the mRNA is going to form, they are going to bind with the 3 prime of the template strand, because 3 prime has OH group free. So mRNA will bind with this template strand with its 3 prime, with its 5 prime mRNA, and then they're going to form, or they're going to make a bond with this template strand. They're going to make a bond with template strand, Once they make a bond, then that bond will be exact similar to the coding strand. Okay, we have seen in mRNA. If this strand gives the information, the mRNA is going to bind with that strand, so that only they will get the equal nitrogenous bases as coding strand. They do not bind with coding strand, they bind with template strand to get the exact copy of coding strand. Okay, we have already discussed. Now here, I'm not going to show the nitrogenous bases. Okay, again I'm going to write this one. I need a space. Okay, let me draw here. This is going to be my last diagram. I'm not going to draw anymore. It has 3 prime, 5 prime, then 5 prime. Here we have 3 prime. This is our coding string. This is our template string. Okay, that we have DNA strand. Now when mRNA is synthesized, apart from these 2 strands, when mRNA is going to synthesize, they first bind with some nucleotide sequences. They first bind with some specific nucleotide sequences. And these nucleotide sequences are present in both the strands. Because this strand, see, we are drawing a straight line, but the actual process are in a coil form, coil structure. In between the coil, the mRNA is going to form. But for our concern, we are drawing like this way. Okay. So first mRNA, it comes and bind with some specific nucleotide sequences. So I'm going to draw that nucleotide sequences as like this. Okay. This way. Suppose this is some nucleotide sequences. This nucleotide sequences is known as promoter. Promoter is a short nucleotide sequences, about 30 to 40 nucleotide sequences, from where the initiation of mRNA takes place, from where the RNA polymerase enzyme comes and bind. Okay, the RNA polymerase enzyme has to come between those 2 strands to initiate the mRNA or to synthesis the mRNA. mRNA synthesis होने के लिए RNA polymerase enzyme का बहुत जरूरी होता है. ठीक है, तो पहले RNA polymerase 3 enzyme कहाँ पे bind होते हैं? RNA polymerase enzyme एक छोटा सा न्यूक्लियोटाइड सीक्वेंस होते हैं, उसमें आके बाइंड होते हैं, एंड दैट न्यूक्लियोटाइड सीक्वेंस इज नोन एज प्रोमोटर. सो फॉर आवर कंसर्न, आई एम शोइंग इन दिस वे. ओके, आई एम शोइंग दिस वे दिस न्यूक्लियोटाइड सीक्वेंसेस. वी कॉल इट एज ए प्रोमोटर. Now promoter, that is nucleotide sequences, is rich in TA data box. Again, go back to mRNA. Again, I made some mistake, silly mistake. mRNA, okay, the 5 prime cap. There I have written data box. There is not a data box. You recorrect it. Okay, I'll come to that. There is not a data box. Okay, उसको काटो. 5 prime में जो cap होते हैं, data box. There is no data box. You उसको काटो उसको. I hope you are getting this. So when mRNA is synthesized, RNA polymerase enzyme has to come, but the RNA polymerase come and bind in short nucleotide sequences where adenine and thymine are rich in number. Okay, that's why we call it as a TA data box. ऐसा sequence में आके bind होते हैं. पहले RNA polymerase enzyme DNA स्ट्रेन का ऐसा सीक्वेंस में आके बाइंड होते हैं जहाँ पे एडिनाइन और थायमिन रिच हो, उसको हम लोग प्रोमोटर बोलते हैं. वो सीक्वेंस को हम लोग प्रोमोटर बोलते हैं. ठीक है, नाउ व्हाट इज प्रोमोटर? प्रोमोटर इज ए शॉर्ट न्यूक्लियोटाइड सीक्वेंसेस, अबाउट 20 to 30 नंबर सीक्वेंसेस होते हैं. सो इट्स ए शॉर्ट न्यूक्लियोटाइड सीक्वेंसेस वेयर एडिनिन एंड थायमिन रिच contain. Okay, so that's why इसको हम लोग टाटा बॉक्स बोलते हैं. मतलब बहुत सारी टाटा थायमिन एडिनिन रहते हैं ये दोनों स्ट्रैंड में, डीएनए स्ट्रैंड में. ओके, उसी को हम लोग प्रोमोटर बोलते हैं. आई होप यू अंडरस्टूड. सो इन प्रोमोटर the RNA polymerase enzyme is going to come and bind. Suppose this is RNA polymerase enzyme. ठीक है, ये एंजाइम को तो आना ही पड़ेगा फॉर द सिंथेसिस ऑफ mRNA, तभी mRNA सिंथेसिस होंगे. दो इन mRNA जब हम लोग डिस्कस किए डायरेक्टली, हम लोग ऐसा डिस्कस किया था ना, ऐसा न्यूक्लियोटाइड बाइंड होते हैं, बट एक्चुअल में ऐसा होता है. So RNA polymerase enzyme has to come and bind. Where it bind? It binds to DNA strand where it is rich in adenine and thymine. Now that sequence of DNA strand which has rich in adenine and thymine, we call it as a promoter. Okay, promoter. That's why Tata box बोलते हैं. Now promoter कहाँ पे होते हैं? Promoter where it is bind. Promoter is bind towards the 5th prime of, I hope it is, you want me to write here? Let me write here. Promoter कहाँ पे bind होते हैं? Promoter, क्या नीचे में लिखना तुम लोग. It binds towards the 5th prime of coding strand upstream. देखो ये चीज़ याद रखना, कभी भी जब भी हम लोग 5 प्राइम लिखते हैं, I don't feel comfortable in Hindi. Okay, let me speak in English. নহলে Assamese. So whenever if you come across a 5th prime, always remember the binding of promoter when it is in 5th prime is always upstream. We call it upstream. Just remember. Okay, so promoter where it is present at the 5th prime of the coding strand upstream towards 3rd prime of template strand downstream. ठीक है, इसको याद रखो again. So whenever there is 3rd prime, 1 minute, So whenever there is 3rd prime, always remember it is downstream. Whenever there is 5th prime, always remember it is upstream. Now promoter is a short nucleotide sequence which comes and which is there. Where? At the 5th prime of the coding strand upstream and the 3rd prime of the template strand downstream. That's you remember. Okay. Now it is a short nucleotide sequences, about 20 to 30 nucleotide sequences which are rich in adenine and thymine. That's why we call it as a Tata box. Remember, important. Okay, so which help in bringing of RNA polymerase enzyme? Sigma bond subunit. Remember, sigma bond subunit are there. It is attached with it. They're going to bring the RNA polymerase enzyme. The RNA polymerase enzyme will come and bind at the promoter sequences which are rich in Tata box. And it is at the 5th prime of the coding strand upstream, 3rd prime of the downstream. Okay. Now, when this RNA polymerase enzymes slide over this strand, template strand, they're going to synthesize a new nucleotide sequences, and that nucleotide sequences is mRNA. It is nothing but mRNA. Okay, when they slide over this strand, they're going to synthesize mRNA along with it. They're going to synthesize an mRNA along with it until and unless they reach a terminator sequences. Again, at the end of the strand, at the end of the DNA strand, there is again another nucleotide sequences where the transcription process is stopped, and that sequences is known as terminator. It doesn't mean with that movie Terminator, it's just named as terminator, where it terminates all the enzymes. Okay, terminator, it contains a terminator. So terminator is attached at the 3 prime of coding strand Whenever there is a 3 prime, that is always downstream. That's you remember. Itu ki hoy keneke hoy bad dia idea nalage. Just mano rakhiba. If I go in, deep, I can go through in very deep, but it is not required. Now just try to understand a basic thing. Okay. So just remember, when it's 3 prime, remember that is downstream. It is attached at the 3 prime of the coding strand downstream and the 5th prime of template strand upstream. That's you remember, and this is very important from entrance point of view as well as for your board exam. Okay, box chala gaya. So there is 2 nucleotide sequences, extra 2 nucleotide sequences. One is promoter and one is terminator. In promoter, it initiates RNA polymerase enzyme, where the RNA polymerase enzyme comes and bind. Once they comes and bind here, they're going to slide over the template strand of DNA. When they slide over, the moment they slide over, they're going to synthesize new mRNA along with it, till it reaches into the terminator sequence. Terminator is also a short nucleotide sequences. So it is attached at the 3rd prime of the coding strand downstream and the 5 prime of the template strand upstream. Okay. Alright, so let me show. So this RNA polymerase tree are sliding over. Try to imagine the images. Okay, slide hoy ase aru template strand or upor phala. Slide hoy ase, then it will until and unless means till it reach here terminator sequence. It's going to synthesis. When they slide over, they are also synthesizing new mRNA strand along with it. New mRNA strand along with. These are mRNA. Okay, these are mRNA. Jitia a RNA polymerase tree tar template strand or upor phale slide kori jabo, tetia notun eta nucleotide sequences add kori jabo. Ei nucleotide sequences to ki mRNA dei ami transcription pori asu. So add kori jabo ketia loge jitia loge hi terminator sequence or goi napai. So yat ahi poa loge stop koribo, kambur ses hoi gol. So once they come at the terminator sequence, it is going to terminate all the enzymes. So enzyme is going to falls off. Now this initiation of mRNA, which subunit help? Yete amar rho factor ahi add hoi jabo. Hey, rho factor ahi add hoi jabo. Rho factor is going to help in the initiation of mRNA. Okay, not only sigma and rho factor. All the subunits are there. Just imagine. Okay, all the subunits are there. If I show all those, the diagram is going to become very complex. So just keep in your mind that all the subunits are attached with it, with its specific role. They are doing its specific role, and they are helping in the initiation of mRNA. That is the process of transcription. For the transcription to occur, there are 3 transcriptional units. Which are 3 transcriptional unit? RNA polymerase enzyme, promoter, and not enzyme. There are 3 transcriptional unit which are there during transcription process. Number 1, promoter. Okay, promoter. Number 2, structural gene. Structural gene will be here in mRNA sequence. I will show next. Then there will be a terminator. So 3 transcriptional unit present hote hai during transcriptional process. One is promoter. It is called as transcriptional unit. Next is structural gene. It is there in mRNA. I will show it. Structural gene. Next is terminator. Okay. I hope you understood. That you remember. Okay. Code attach hoy. Let's revise. So for the transcription to occur, the RNA polymerase enzyme has to come and bind to the template strand of the DNA. First of all, now what is template strand and coding strand we have already understood. So RNA polymerase comes and bind to the promoter sequence, which is rich in adenine and thymine. Okay, so that's why we call it as a Tata box. RNA polymerase enzyme will come and bind to the promoter sequence. Once they come and bind to the promoter sequence, it will slide over the template strand. When they slide over the template strand, Along with, they also synthesize a new mRNA with it, okay, until it reaches into the terminator sequence. They're going to synthesize mRNA. When they reach the terminator sequence, they're going to false off. All the enzymes and subunit, they're going to false off. Then a new mRNA has been synthesized. Where promoter is attached at the 5th prime of the coding strand upstream, 3rd prime of the template strand downstream. Whenever the word's upstream, that means 5th prime. Whenever the word is downstream, it means 3rd prime. That's, you remember why, how, when, no need. Just remember, if I go in deep, you will get confused. And then the terminator is the sequence where the process, that is the initiation of mRNA or synthesis of mRNA, stops. And it is a sequence which is attached at the 3rd prime of the coding strand downstream, 5th prime of the template strand upstream. ठीक है, इसको याद रखना. So that is the process that takes place. ठीक है? Now coming back. Okay, so let me show you rough again. This 3 prime, it has 5th prime, the 5th prime, 3rd prime. This is our coding, template strand. This is our coding strand. So here we have, suppose A, T, C, G, T, C, G, A, T. So next to it, the DNA strand A, T, A, G, C, A, G, C, T, A. So this is, we have a genetic code, nitrogenous bases. So the mRNA, when they form, so try to relate this diagram with this, okay? Enzymes are there, terminator, promoter are there. So the new strand, that is mRNA, is going to come here with this 5th prime of it, because 3rd prime has the OS free here. So 5th prime will make a bond with it. So they're going to synthesis, they're going to make a bond with this to get the exact copy of coding strand. This is our coding strand. ओके मेरा mRNA वो क्लास में साइड मैंने यहाँ में 5 प्राइम दिया है, उसको 3rd प्राइम करो, यहाँ में 5, हाँ यहाँ में 5 प्राइम करो mRNA में जाके, ठीक है. So see, so adenine will bind with thymine, but this is mRNA, so thymine will replace by uracil. इसका प्रॉब्लम नहीं है, कितना समझाऊँ एक ही बात को. Yes, so we got the exact copy like this. So this is our genetic code. Now this is our mRNA, the first synthesis strand of mRNA. Now this mRNA strand, the strand which has been synthesized for the first time, we call that strand as hnRNA. hnRNA, heteronuclear RNA. Okay, hnmRNA or hnRNA, ऐसा भी बोल सकते हैं, hnRNA. So this what is hnRNA? It is a heteronuclear RNA. The first strand which has been synthesized from the template strand of the DNA. That strand we call it as a hnRNA. Now there is a problem, right? So they have all the genetic code along with it. But we know the codes are read in a triplet form, right? But this strand also contains some non-triplet code. Okay? That means non-coding. ट्रिपलेट को हम लोग कोडिंग स्ट्रैंड बोलते हैं, ठीक है, कोडिंग. सो along with the coding, they also contain some non-coding part. So non-coding part has to remove. Okay, to remove non-coding part, the strand undergo splicing process. Now with the help of enzyme endonuclease, the strands are cut into pieces. We have already discussed, right, this one. Again we are discussing here. The strands are cut into pieces. Okay, now next thing, the coding are known as exon. Non-coding are called it as a intron. Okay, coding mane juntu codes triplet form of thakibo tak ami coding boli kom, non-coding juntu codes triplet form of na thakibo tak ami non-coding boli kom. Aru tare coding on ami exon bolio kom, intron non-coding o intron boli kom mRNA khetro. ঠিক আছে, so along with this strand it contains exon. Suppose this is exon 1, exon 2, exon 3. Here we have intron 1, intron 2. So we need to remove intron, that is non-coding part. So to do that, an enzyme act on it, endonuclease enzyme. The endonuclease enzyme is going to break all these strand into pieces. Okay, so here we have exon 1, exon 2, exon 3. So intron color alag alag ho gaya. So here we have intron 1, intron 2 and so on. So we will join only exon part, ठीक है? Exon part को ही join करेंगे हम लोग, because exon are coding. So this joining of exon are done by with the help of enzyme ligase. Okay, so we have now only exon part, that is coding part. Now that is our mRNA. We will not call it as a hnmRNA. Now we will call it as a mRNA. But this mRNA is unstable. Though RNA is unstable only, but to make it little bit stable for the translation process, a cap has been added at the 5th prime and tail has been added at the 3rd prime. Now prime is very important. कौन सा prime में क्या add करते हैं इसको याद रखना बहुत जरूरी होता है. You have to remember. I don't want to rub this diagram, but I have to do. Let me show here. Okay. So this mRNA strand. Now what happened to make it stable? A cap and a tail has been added. The cap is added at the 5th prime of it. This cap contains methyl guanine, not Tata box. Okay, you make it correct this. This cap contains methyl guanine, and the tail contains, where tail is added? Tail is added at the 3rd prime of the mRNA strand. The tail contains many adenine, so we call it as a polyadenine or polyadenylate tail. Polyadenylated tail. Okay, we have already seen that. So now we have a complete structure of mRNA which is ready for the translation process. Okay, now the first sequence of mRNA contains here start codon. This first sequence of mRNA contains start codon. Example of start codon, this is start codon AUG. Remember, AUG is the first start codon. Then towards the last part of the mRNA strand here, it contains stop codon or nonsense codon. I hope it is visible. Example, UAA, UAG or UGA. It could be any one of these. UAA, UAG and UGA. These are stop codon. The role of stop codon we will see. Okay. So the cap contains methyl guanine, not Tata. I have written Tata in mRNA, so recorrect it. Methyl guanine. The tail will contain many adenine, so we call it as a polyadenine. And the first strand of mRNA contains start codon, that is AUG. And the last strand of mRNA contains stop codon, UAA, UAG or UGA, any one of those. So from start codon to stop codon, the strand of mRNA we call it as a structural gene. Okay, structural gene. Now the transcriptional unit, which are our transcriptional unit, they are promoter, structural gene and terminator. These are our transcriptional gene, transcriptional unit. Understood? Very easy, very interesting, very easy. Alright, let's again revise. So, for the mRNA to synthesize, the RNA polymerase enzyme helps a very important role. It does, it has a very important role. RNA polymerase enzyme. So types and all we have discussed, subunit. So DNA, the mRNA is going to carry information from the DNA strand only. So the strand which is going to give the information to mRNA, we call it as a coding strand, and the other strand will become a template strand. Now mRNA will make a bond with template strand to get the coding strand. It doesn't make a bond with coding strand to get a coding strand. Okay, that's you remember. So mRNA is going to make a bond with template strand. So first RNA polymerase come and bind to the promoter sequences which is rich in thymine and adenine. Okay, once they come and bind here, they're going to slide over the template strand. When they slide over the template strand, they're going to synthesize all the mRNA, new nucleotide from the template strand of the DNA to get the exact copy of coding strand. So the process will continue till it reaches into the terminator sequence, where all this synthesis stops and all the enzyme and subunit falls off. Once the mRNA is synthesized, the first mRNA we call it as a heteronuclear mRNA. Let me write mRNA, otherwise you will get confused. The heteronuclear mRNA. Now this first synthesis of mRNA strand contains some coding and non-coding part of it. The coding is known as exon, the non-coding part is known as intron. And we need, that means our body need only exon part. So intron part has to remove. To remove intron part, it undergoes splicing process. With the help of enzyme endonuclease, the strands are cut into pieces where the exon and intron separates. Then with the help of ligase enzyme, only the exon part gets synthesized, but this strand of mRNA is unstable. To make it stable, a cap has been added at the 5th prime of the mRNA strand, which has a methyl guanine in it, and the tail has been added at the 3rd prime of mRNA, which is rich in adenine. That's why we also call it as a polyadenine or polyadenylated tail. And the first strand of mRNA contains start codon, example AUG, and the end part of the mRNA strand contains stop codon or nonsense codon, UAA, UAG and UGA. Any one of those will contain here. So starting from start codon to stop codon, we call it as a structural gene. So that's it. That is the process of transcription. Okay. So the process is completed in 3 steps. Number 1 is initiation. You write it by your own. Step 2, elongation. And step 3, termination. So your notes should be with respect to the diagram. Diagram is important. Okay, so the process, the steps of transcription are completed under 3 processes. First is initiation, where it will initiate RNA polymerase enzyme. How it initiate, we have already discussed. Then elongation, the RNA polymerase enzyme will elongate towards the template strand. When they elongate, they will synthesize the mRNA along with it. Then termination, finally it will terminate all the enzymes and subunit. They will going to false off the mRNA synthesis. That's it. And we have 3 transcriptional unit. They are promoter, this one, structural gene, that one, then terminator gene, that one. Okay, terminator, not gene, terminator sequence. Alright, so write it by your own sentence. This is the diagram, all those. Okay, so I hope you understood. Next class we will see translation process. If you have still some confusion, you can ask me. Okay.